Okay.
Yeah.
Hi, thanks for inviting me to give a talk today. My name is Liz Hetherington, and I'm a postdoc researcher in Anella Choi's lab at Scripps Institution of Oceanography in California. Today, I'm going to talk about some of our work on Siphonophores, which are pictured here and are a clade of hydrozoan jellies. So first, I'm going to talk about some of our broader work on deep pelagic community composition and the roles that gelatinous animals like Siphonophores play or occupy in these habitats. And then I'll focus on two diet studies on Siphonophores. One is a literature synthesis, and the other is our current work on looking at Siphonophore diet using stable isotope analysis. So this is just a little overview of my talk today. And over the last several decades, paradigms around the roles of gelatinous zooplankton in food webs have shifted. And we can see this by looking at the number of publications on scyphozoans, or true jellies, these large medusae that we typically think of as true jellies. The research interest has increased, and we see the research interest has generated different thinking about gelatinous animals. So we know now that they're important predators, prey, mediators of particle flux. But most of the work on gelatinous zooplankton has focused more on the scyphozoan medusae and not on other groups of gelatinous animals. Keep in mind that the term gelatinous zooplankton is an umbrella term that's used, but actually represents a diversity of animals from several phyla. And we haven't seen a similar increase for some of these groups, increase in interest. So if we think about Siphonophores, they're relatively understudied compared to other gelatinous animals, even though they're quite ubiquitous in zooplankton communities. And part of the reason that we don't know a lot about their roles in food webs or ecosystems is because they're particularly fragile and they're difficult to collect. And these sampling challenges can really affect our perception of their contribution to community composition. And we illustrate this in a paper that was published just last week in Frontiers in Marine Science. So in this study, we sampled the pelagic community using 3 different methods: 2 trawls, so 1 a mock nest, 2 a modified Tucker trawl with an insulated cod end, and then the 3rd method was a visual survey using a remotely operated vehicle video transects, and this was conducted in Monterey Bay. We sampled the community at 3 depths: 150 meters, 350 meters, and 750 meters. And these 3 pie charts here show the percent relative abundance of broad taxonomic groups that were collected using these methods. And it's pretty clear that there were some major differences where the mock nest collected mostly crustaceans, not a lot of gelatinous animals. The Tucker trawl was more evenly distributed among organisms. And then the ROV video captured much more gelatinous taxa and fewer crustaceans. So, and in addition, the ROV was the only method that captured the most physically delicate specimens. And these are animals like lobate ctenophores, larvaceans pictured here, or our Physonex Siphonophores. If you're not familiar with Siphonophores, they're an order of colonial cnidarians. They're divided into 3 suborders, but I'm going to specifically talk about the Chalicophorans and the Physonex primarily because most Siphonophore species are in these 2 suborders. And the main structure of Siphonophores is that they have a pneumatophore, or a little float used for buoyancy, and then they have these swimming bells or nectophores that they use for locomotion. And then they have different types of zooids on this area called a cyphosome. So this cyphosome has digestive zooids, reproductive zooids, and like other cnidarians, they have tentacles with stinging cells. So you can see this here. They're tentacles that they use to capture prey. And they may have served some interesting roles in the food web because unlike most other animals, they're not gape limited. So they don't have one mouth, rather they have a bunch of stomachs along the length of the colony. First, we wanted to know, what do siphonophores eat? What's known about their roles in food webs? So we conducted a literature synthesis of all siphonophore predator-prey interactions. So this was, we took both quantitative and qualitative diet data from the literature and created these food web graphics based on epipelagic and mesopelagic interactions. And we separated these because they're conducted using different methodologies. Epipelagic interactions were primarily from gut contents analysis, from specimens collected blue water diving, and mesopelagic were primarily from remotely operated vehicle observations. And in these very busy food web figures, I just want to point out a few things. So first, siphonophores are active predators that are preying on a wide diversity of zooplankton and micronectin taxa in both the epipelagic and mesopelagic. And these predator-prey interactions look different between the shallow and deep ocean. And lastly, there's very limited data on predation on siphonophores, but from the literature, it's clear that they're not trophic dead ends, and they are preyed on by several higher trophic level taxa. And this work was recently published in L&O Letters. We also, in this synthesis, took the quantitative diet data and compared it across siphonophore suborders, and we see that Cystinex, so the Portuguese man-o'-war and a few other species, are primarily fish eaters. Chalicophorans are primarily consuming small crustaceans like copepods, and Physinox consume a lot of crustaceans as well, but have more varied trophic niches or dietary niches than Chalicophorans. And there appears to be a lot of species-specific specialization in the Physinox species. And this was based on 28 species of Siphonophores. We took the predator siphonophore prey interactions and created predator-prey bipartite networks using the bipartite package in R. And we created networks for the epipelagic again and the deep pelagic, and compared several network parameters to compare food web topology between siphonophore prey interactions in the epi and deep pelagic. So we compared things like diversity and connectance or a complexity of these networks. And I just want to point out two things that we found that were quite interesting, was that specialization was higher in the deep pelagic network, and nestedness was lower. So this is indicating that siphonophores in the deep pelagic are more specialized, and these specialist diets aren't just subsets of generalist diets, that they're carving out these distinct ecological niches. So that synthesis was based on a limited number of siphonophores that were collected using different methods and across different ecosystems. So our next task was to sample siphonophores from one community to be able to examine diet across the water column. So we collected siphonophores off Central and Southern California on several cruises, and we collected specimens using multiple methods, so blue water diving, remotely operated vehicles, and tucker trawls. And we sampled tissues for stable isotope analyses. And this was to get a different perspective of diet. So unlike gut contents analysis, it provides a longer term picture of diet or trophic ecology. We can use biochemical tracer techniques like stable isotope analysis to examine diet integrated over time, because nitrogen isotope ratios, or delta and 15, increase in a somewhat predictable manner with each trophic transfer as you move up the food web. So we can use these nitrogen isotope values to estimate the trophic position or the placement of animals in the food web. And we did this with our the Siphonophores that we collected. We estimated trophic position, and we do see that Physonex overall have higher trophic positions than Chalicophorans, and their trophic positions also vary more. So there's a wider range in trophic position. They're feeding across more trophic levels. But we didn't find a difference in trophic position with collection depth, although the highest trophic position estimates were from deep pelagic Physonex species. Our isotope data are therefore suggesting that some Siphonophores, so Chalicophorans and Physonex, are feeding on small crustaceans like copepods. Then there are other species that are feeding on larger crustaceans, some small mollusks. And then there are other Siphonophores, primarily Physonex, some of these larger Physonex that are feeding on fishes, gelatinous animals, higher trophic level species. So these biogeochemical analyses in our literature review show that just one clade of gelatinous animals occupies multiple trophic levels or niches in the food web, in pelagic food webs. And keep in mind that most ecosystem or food web models either don't include gelatinous animals or include them as one functional group. So from here, we are actively working on our isotope manuscript, and also have a metabor coding manuscript that's coming out soon. And we're excited to expand upon what we know about Siphonophore diets into these biochemical and molecular frameworks. And from here, we can ask more detailed questions about the drivers of Siphonophore diets, and particularly how their diets change with vertical structure through the water column, and ultimately understanding how energy flows through the gelatinous components of the food web will provide a better picture of food web interactions, energy flow, and how these species interactions shape food web structure. And with that, I'm happy to take any questions. I just want to thank all of my collaborators for their input over the years. And yeah, with that, I'll take any questions.
Thanks, Liz. That was so great. Remind me to never have a poster presentation next to you, because your figures are amazing.
Oh, thank you.
No, it's great. It's really good work. And all the papers, I saw the paper that came out last week too, and it was, yeah, it's so interesting. It's just right down my alley kind of. So yeah, it's lovely. Any questions, people? Maybe I start. So you just focused on like West Coast of US, but do you think these results will be different in different regions?
Yeah, that's a good question. It does seem like some of the Siphonophores have pretty are specialists, and some are generalists. So I might think that some of the specialist diets would be pretty similar, but we really don't know. We have some comparative analyses for a few species that were collected off the north, like in the Northeast, off the in the northwestern Atlantic, so off the northeastern coast of the US.
Yeah.
But just for like 2 or 3 species, and the results are similar between those, but we really don't have a good idea of that.
Yeah, fair enough. Yeah, because I mean, I followed other voyages last year. I mean, was it last year or the year before, even the exports voyage? And just looking at a picture of micronecton from the mesopelagic, it looks pretty similar what we get out of the Southern Ocean. So I'm wondering, I mean, they're probably generalist, but how, really regional populations are, or if it's really, mesopelagic is really more uniform than we thought. But yeah, it might be, that specialists are really just, would change between regions, but generalists would be the same.
Yeah, and another big thing is that the Siphonophore depth distributions aren't really well characterized.
Yeah.
So a big part of drivers of diet is prey availability, and to kind of answer some of those questions, we really need better information about their depth distributions. A lot of Siphonophores, perhaps most of them, are migrators, so that kind of getting more information about the vertical distribution will probably elucidate some of that.
Yeah, definitely. Katie, do you have a question? Oh, yeah.
Yeah, hi, thank you so much, Elizabeth. That was really interesting. I had a question about your sampling methods. I noticed that your team, at least in this study, was not using acoustics. And to kind of continue with what you were just saying about the depth distribution, I wondered if that is a consideration that you'll use in future samplings, and how to integrate acoustics in the future if you decide to do that. That's a great point. It's something that, at least for the last several cruises we've gone on, we have collected acoustic data. I personally am not, haven't been involved in that, and we haven't necessarily related it to Siphonophore abundance, but certainly it can be useful and can be really well integrated, especially because trawl surveys typically collect Chalicophorans Siphonophores because they're not as fragile, but they don't have a gas bladder, so those aren't the ones that you're seeing with acoustics. But the really delicate Physonic ones, those are the ones that have the gas bladders, and so they're not collected by trawls, but they are collected or seen with acoustics. So yeah, there's really large potential for that, and I think combining acoustics with some of the visual methods. So some of our team members are working on vertical distributions from ROV surveys as well. So I think combining some of those methods will be really powerful for the Physonics. Yeah, very challenging though.
Yes, I'm sure.
Yeah, okay. Great, thank you.
We have a question in the chat. Elva asked, have you characterized eDNA to detect the presence of Siphonophores? eDNA, if you have used eDNA.
Oh, have we used eDNA?
Yeah.
No. Yeah. So not to my knowledge. Somebody else, Alejandro Damian Serrano at Yale, he did DNA work on the gut contents of Siphonophores. So looking at Siphonophore prey, so what they're eating, but to my knowledge, I can't think of any eDNA studies that specifically, targeted or discussed Siphonophores. But yeah, that's definitely a method that could also be used to, especially for some of the really delicate taxa.
Awesome. Any